everybody. Today's topic is the ovary and oogenesis. In the video on female reproductive system, we have learned some few points about the ovary, and today we are going to discuss it in detail. The ovaries are covered on the outside by a layer of simple cuboidal epithelium, and underneath this layer there is a dense layer of tissue, and this is known as tunica albuginea. Germinal epithelium, the first one, then the next one is tunica albuginea. The inside part of the ovary is distinctly divided into two parts: the outer cortex part and the inner medulla part. The cortex appears very dense and granular in structure, and this is due to the presence of numerous ovarian follicles. They are in various stages of development. Each of the follicle contains an oocyte that we are going to discuss. And this oocyte is a female germ cell. This medulla part, it is made out of loose connective tissue, and it has blood vessels, lymphatic vessels, nerve fibers, etc. The ovaries are the primary female reproductive organs or female gonads. There are two ovaries. Each ovary is a solid structure about the size and shape of an almond. The length is around 3.5 cm, breadth is around 2 cm, and the thickness is around 1 cm. Where are they located? They are located in a shallow depression. The name of the depression is ovarian foci. One on each side of the uterus and in the lateral part of the pelvic cavity. They are loosely held in this depression part by some ligaments known as peritoneal ligaments. The ovulation occurs in one of the two ovaries generally, and the process is known as oogenesis. This process starts with the primitive germ cells or primordial germ cells. These primitive germ cells appear at caudal part of yolk sac. Let us see that. This is the embryo, and this part is known as the yolk sac. And these primitive germ cells, they appear here in the skin of yolk sac at the second week of gestation. They are also called PGC. Then these PGC, they migrate to reach the developing gonads, that is, testes in males and ovaries in female, at the 5th week. So the appearance of PGCs at second week, and they migrate to ovaries at the 5th week. In female, this primitive germ cells is present in the cortex of the ovary. They multiply and named as oogonia. So we'll start with oogonia. And singular is oogonium. Majority of oogonia continue to divide by mitosis and form more oogonia. But some oogonia, they give rise to primary oocytes and enter the prophase of meiosis 1. This part is very important. So this is primary oocyte. This is the nucleus. This is cytoplasm part. And they are arrested at prophase 1 of meiosis. By 5th month, number of germ cells, these oogonia and primary oocytes, they reach around 7 million in the ovaries. And then cell death begins. This process is known as atresia. By 7th month, majority of oogonia and primary oocyte degenerate. So this whole process is before birth till this primary oocyte. At the time of birth, all the surviving primary oocytes are surrounded by a single layer of cells known as follicular epithelial cells. And at birth, this number of primary oocytes had reduced to approximately 1 to 2 million. And the primary oocyte surrounded by a layer of follicular epithelial cell is known as primordial follicle. The primordial follicles always form the majority of follicles in the ovary. They are typically dormant in nature because the primary oocyte is arrested at prophase 1. They are developed at the fetal stage, and this is the first stage. Second stage is the primary follicle. Till puberty, these primordial follicles remain in dormant stage, and after puberty they develop to primary follicle. So till this stage it is before birth, primordial follicle at birth, and at puberty it is primary follicle. The primary follicles are developed from primordial ones under the influence of hormones. They are comparatively larger and surrounded by layers of cuboidal granulosa cells. These follicular epithelial cells that you have seen before, they are also known as granulosa cells. Here they are flat granulosa cells, here they are cuboidal granulosa cells. And a very significant thing in primary follicle is the development of a layer between the primary oocyte and granulosa cells. They are known as zona pellucida. So here in primary follicles, in comparison with primordial follicle, there are two distinct features. One is cuboidal granulosa, another is zona pellucida. At puberty, the number of primary follicles dropped to about 40,000. Here it was around 1 to 2 million. These granulosa cells are the receptor of FSH. FSH is follicle stimulating hormone, and that triggers the start of ovarian cycle. And from these 40,000 follicles, around 400 to 500 only ovulate in the whole reproductive life of a female. And this is the total supply for a lifetime. Unlike sperms keep on generating in the body till the end of the male reproductive cycle. I have shown the stages from oogonia to primary follicle. In between stages I have explained here. The primary follicles which survive then develop to secondary follicle. In the secondary follicle you can see the layers of granulosa cells. It is known as the stratum granulosum. In the secondary follicle, the zona pellucida is more visible. This layer is made out of glycoproteins. The outside layer of stratum granulosum is known as the basal lamina, and outside these theca cells start generating. If the secondary follicles survive the follicular atresia, then tertiary follicles are developed. The characteristics of this tertiary follicle is the presence of this antral cavity. It is a fluid-filled cavity. A mound of follicular cells are formed on which this oocyte resides. This mound is made up of granulosa epithelial cells, and the name is cumulus oophorus. The theca layer has developed into two distinct theca interna and theca externa. In response to the rise of FSH, that is follicle stimulating hormone, the antral follicles begin to secrete estrogen and inhibin. This inhibin has a negative feedback effect on FSH. Follicles that have fewer FSH receptors, they won't be able to develop further, and eventually only one follicle will be available for further growth. This remaining follicle will grow very quickly to become Graafian follicle. This is actually a very large tertiary follicle which will go to the path of ovulation. This primary oocyte divides into a secondary oocyte and a polar body that we will show later. So a Graafian follicle has a secondary oocyte. Here this columnus oophorus that helps to degrade and release the secondary oocyte at the time of ovulation. This antral follicle, granulosa cells, and theca interna and externa. This layer of granulosa cells, they have microvilli and known as corona radiata. This corona radiata and this secondary oocyte is the sign of Graafian follicle, which is ready to just come out of the ovary. Here these are the various stages from primary follicle, secondary follicle, further growth gives tertiary follicle and a big Graafian follicle. It is so big that on the outside of the ovary you can see a blister here. Next, the suitable combination of hormones rupture the follicle and the egg or ovum comes out. Next stage is the corpus luteum. This corpus luteum develops from this ovarian follicle during the luteal phase, when the release of the secondary oocyte from the follicle happens. The secondary oocyte traverses the fallopian tube and this luteum remains in the ovary. It is an important part for establishing and maintaining pregnancy in females. It secretes a hormone known as progesterone. This is responsible for the development and maintenance of endometrium, that is the innermost layer of the uterine cavity. But when the egg is not fertilized, the corpus luteum stops secreting progesterone and degenerates after around 10 days in humans. It continues degenerating and forms corpus albicans, which is a mass of fibrous scar. And outside the ovary, the uterine lining, that is the endometrium, is expelled through the vagina and the menstruation cycle starts. Now let's see the effect of hormones on folliculogenesis, that is the development of follicles. It is controlled by the endocrine system and 5 hormones take part here. The gonadotropin releasing hormone known as GnRH. Gonadotropic releasing hormone. It is secreted by the hypothalamus. GnRH stimulates the release of FSH, follicle stimulating hormone, and LH, luteinizing hormone, from the anterior pituitary gland. There is a large amount of GnRH just before ovulation. In females, this FSH initiates follicular growth, specifically granulosa cells growth. The LH surge, that is secretion of large amount of LH, triggers ovulation and development of the corpus luteum. This graph is divided into 2 sections, follicle phase and luteal phase, from 0 to 14 days and 14 to 28 days. And this part is the ovulation part. PF, SF, TF and GF, they are primary, secondary, tertiary and Graafian follicles. When theca cells are formed in the tertiary follicle, then the amount of estrogen increases sharply. These theca cells, they derive androgen, then the granulosa cells convert it into estrogen. Theca cells, androgen, and granulosa cells convert androgen to estrogen. As more estrogen is secreted, more LH receptors are made by the theca cells. Then this causes LH to spike sharply, and this spike causes ovulation. Here see, from 0 to 14D, this is estrogen. With this surge, the surge of LH happens. Following ovulation, LH also stimulates the formation of the corpus luteum. The amount of estrogen has dropped after ovulation. Inhibin is secreted by corpus luteum and it contributes to this diminishing FSH. After ovulation, corpus luteum is formed and it starts secreting progesterone. As we have said, it is very essential for maintaining the endometrium for both maintenance and development. This is very clear from the graph that just before ovulation, the surge of FSH, E and LH happens, and after ovulation, the surge of P happens. Now let's draw the flow diagram. We'll start from oogonium, and by mitosis it forms primary oocyte. Here 2N and 2N, they are diploid, and the primary oocyte is arrested in prophase 1. This whole part is before birth. Next stage is after puberty. Here I should write prophase 1 of meiosis. Next, meiosis continues and secondary oocyte is formed. Here this is haploid, only N. This meiosis is arrested in metaphase 2, it is not complete. And the cleavage of primary oocyte is not symmetric here. This is the cleavage part. So this part will form a polar body and this part secondary oocyte. Here the amount of cytoplasm differs. Here you have seen this secondary oocyte is arrested in metaphase 2 and this will remain in this stage till the fertilization. So here sperm will come and fertilization will take place. At the time of fertilization, the process meiosis will get completed and the fertilized egg will form. Here it is again 2N because it is fertilized now, and a polar body again will generate. This is second polar body. These polar bodies serve to eliminate 1/2 of the diploid chromosome set produced by this mitotic division, leaving behind this haploid secondary oocyte. Sometimes this first polar body divides into one more polar body and altogether 3 polar bodies happened in this cycle. Generally these polar bodies degenerate. But the egg cell and a polar body may both get fertilized by separate sperm. But even if this fertilization occurs, further development won't happen because of this insufficient amount of cytoplasm. They don't have proper amount of nutrients to feed the embryo. Our next video will be on spermatogenesis. If you like this video, please subscribe to our channel and thank you for watching.